Okay, so we left off yesterday, or the last video, talking about the stimulation of glycogen breakdown by the signal molecule epinephrine. And so I talked about how one signal molecule can elicit a huge response, many more molecules activated than for that one molecule. And so this is called signal amplification. And so the signal is amplified as we go along. Signal amplification. All right, and so you get a big response. So many other signaling pathways regulate the synthesis of enzymes and other proteins, usually by turning genes on or off in the nucleus. So therefore the genes will stay off unless a signal molecule binds to the surface of the cell. And so the final activated molecule may function as what we call a transcription factor, because transcription is the first stage in which you take your DNA and start to kind of quote unquote read it to make proteins. And proteins are going to give a cell its traits. So if we look here at this picture here, so this is just a review again. Here's your reception. Here is a molecule, let's say it's a growth factor that's going to stimulate growth, okay? And so it binds to the receptor. The receptor, remember, changes shape. That's the reception. That shape change signals a relay protein that could be a second messenger like CAMP, cyclic AMP, which then activates another molecule by phosphorylating it, which then activates the next molecule by phosphorylating it, and so on and so forth. That's why it's called a phosphorylation cascade. That's the transduction part. And then eventually at the end of this pathway, you get an activated molecule that's going to be instrumental in eliciting your response. But look at here, instead of something happening here in the cytoplasm where it activates a molecule that gives you your response in the cytoplasm, this activated molecule enters the nucleus. So this guy is activated, and it's a phosphorylation cascade, so often activated by being phosphorylated. It enters the nucleus, and so it goes through the nuclear pore. And notice here you have an inactive transcription factor, but when this little guy comes in, it activates it again by phosphorylating it. So you see here we have our phosphate group right here. So this is phosphorylated. So why is it active? I'll put here phosphorylated. And now if you look at the picture, what does this active transcription factor do? It binds to the DNA, so binds to the DNA, and what it does is it starts, notice here it says gene, so remember a gene is one part of your DNA. So from here to here is the gene, so it goes to the start of the gene, binds to the gene, and notice here it makes mRNA. I'm going to put here copies instructions to make proteins from DNA onto mRNA. So mRNA is the instructions. So this contains the instructions. So it copies those instructions, because remember, DNA does not leave the nucleus. And we're going to make a protein, and proteins are made outside the nucleus. Where? Outside the nucleus, and the little guy is called the ribosome. So this is outside of the nucleus. And so this is going to go to the ribosome and make a protein, which then will give, which would then lead to your response. Or sometimes in genetics it's called a trait, because that protein will do something like be an enzyme that's going to do a certain job, and then that gives you a certain trait because you have that characteristic. All right, so sometimes at the end of that phosphorylation cascade, it acts as a transcription factor and turns on genes. So then let's wrap this up here. Some just minor details here. It says fine tuning of the response. Multi-step pathways have two important benefits. So this is definitely a multi-step pathway to elicit a response. The first one is being able to amplify the signal. Amplifying the signal, and thus the response. That's like in this figure over here, where we get, from one, we get many molecules made, and then contributing to the specificity of the response. So we can get very specific responses in the cell. And so just a quick little review here. I didn't make any fill in the blanks here. So the signal amplification. So these two here, we're going to look at these. So this one's right here and this one's right here, just to see that. So this is 1 and 2. The enzyme cascades amplify the cell's response. At each step, the number of activated products is much greater than in the preceding step. And so that is this figure right here, all right, that we talked about in the previous video. And the specificity of the signaling. Different kinds of cells have different collections of proteins, so not every cell is the same. These differences in proteins give each kind of cell specificity in detecting and responding to signals. So therefore, not every cell responds to every signal. And so therefore, a hormone can travel through the bloodstream and only affect certain cells of the body and not every single cell, because not every single cell has the right proteins, the right receptor proteins. So the response of a cell to a signal depends on the cell's particular collection of proteins. So sometimes there's what we call pathway branching and crosstalk, further help cells coordinate incoming signals. And so that's what this picture is showing you here. So if we look here, we have 4 different examples: cell A, cell B, cell C, cell D. So let's look at this. So cell A, it says pathway leads to a single response. So here's your reception. Signal molecule binds to the receptor in the membrane, activates a relay molecule, which then activates another, and so on, and eventually you get your response. So that's pretty simple, leads to a single response. Cell B, you can get different responses. And so cell B, the signal molecule binds, it activates a relay molecule, and then at the end activates different molecules, which then lead to different responses. This right here is often what happens. This could be a receptor, are tyrosine kinase receptors. How do I know that? That was one of the receptions. Tyrosine kinase reception leads to multiple responses. Whoops, I'm off the multiple responses. And so if you look back at the different kinds of receptors, there were gated ion channels, there were G protein linked receptors, and there were tyrosine kinase molecules, and so that leads to multiple. Cell C says crosstalk between 2 pathways. So notice here we have 2 different receptor molecules, 2 different signal molecules. So they bind here to the receptor, and it activates different protein molecules. And then notice here that this one, it says activation or inhibition. So this molecule here stimulates this molecule, which leads to a response. This molecule can either also stimulate this molecule and lead to the same response. So 2 different molecules can lead to the same response. So therefore your body can, different parts of the body can release different signal molecules that may end up having the same responses in the cell that another molecule does. Or it can actually inhibit it and stop that from happening. So this signal molecule kind of would cancel out this one. And that's a possibility as well. So that's the crosstalk. And then cell D. Cell D here, we have a different type of receptor. So it says a different receptor leads to a different response. So again, if you notice these three here, 1, 2, and 3, have the same shape and the same signal molecule. All right, so but they can activate different types of proteins inside here. Now we have a different receptor, so in that same membrane we can have different receptors that will activate different molecules that will lead to a different response 5. So therefore, from that same signal molecule, you can get lots of different types of responses depending upon the protein embedded in the membrane and what it activates. Okay? So that's the specificity of cell signaling. So the different collections of proteins, as we're talking about, the response of a cell to a signal depends on the cell's particular collection of proteins. So if a cell has this collection of proteins, it'll be different than a cell that has this collection of proteins, this collection, or this collection. And so therefore, the same signal molecule can have different types of reactions in different types of cells. Okay. This part right here, this is exciting for you. You get to cross off, all right? So this is not on our test. This is not on the AP test. I just forgot to delete it, so we're just going to cross that part off. And lastly, determination of the signal. We don't want this signal to last forever, this communication. So inactivation mechanisms are an essential part of cell signaling. When when the signal molecules leave the receptor reverts to its inactive state. So if we go back to, like this here picture, here's the signal molecule. When the signal molecule comes back off, it goes back to inactive and none of this happens. And so we have to wait for the next molecule to come. And so that is the end. I am going to have you on the back side of this, we're going to write down a little bit here that something that has to do with this, and this is called apoptosis. And so let's apoptosis. So what is this? Let's just write some bullets here. Is a programmed or controlled cell suicide. So the cell kills itself. Let's write the next one. Components of the cell are chopped up and packaged into vesicles that are digested by scavenger cells. Apoptosis prevents enzymes from leaking out of a dying cell and damaging neighboring cells. So what triggers this? Can be triggered by what? One, and this is how it relates to this chapter, an extracellular, that means outside the cell, death signaling ligand. So a molecule outside the cell can bind to a receptor protein, which triggers this death process. 2, DNA damage in the nucleus. So, and this makes sense because if you've got damage in the cell, the DNA controls everything. And so if the DNA is damaged, then you've got a damaged cell. You probably don't want to let that cell live and divide and reproduce more cells that there's something wrong with. Or 3, protein misfolding in the ER, the endoplasmic reticulum. So if a protein isn't folded correctly and doesn't have the right shape, it's not going to have the right function, and so therefore there's something wrong with it, and so that can trigger apoptosis as well. So where this come from, apoptosis is a good thing. It can be a good thing. So apoptosis evolved early in animal evolution and is essential for the development and maintenance of all animals. We, in early on in development, have skin. We have cells in between our fingers. They're like webbed between our fingers and also our toes. In development, while we're still inside of our mother, those cells undergo apoptosis and die, leaving us with free-moving fingers and free-moving toes. So that's an example of it being essential for the development in humans. It also can be a bad thing if it happens when it's not supposed to. So apoptosis, they think, may be involved in a couple of diseases. And one of those is Parkinson's and Alzheimer's. And they also think that interference with apoptosis may contribute to some cancers. So when a cell is supposed to die and it doesn't, should be, killed because there's something wrong with it, which cancer cells have something wrong with them and they continually divide uncontrollably. So that can contribute to some cancers, they think. So I wanted to show you a picture here of apoptosis. Apoptosis in a mouse. And so here's the mouse paw early on in development, and here are the cells. They have tissue in between their digits right now, just like I talked about us having that. And here's the cells undergoing apoptosis, and eventually you see how now there's space between the digits. And so that is an example of a normal part of apoptosis during development. What's going on cellularly, this is an actual picture of apoptosis of a human white blood cell. So this is the normal white blood cell, and this is when it's undergoing apoptosis. And so notice here, it looks like it has all these little bumps. They call them blebs. And so it's shrinking and forming these membrane-bound little vesicles. And so that's when we wrote here, components of the cell are chopped up and packaged into vesicles that eventually are digested by scavenger cells cells. Those scavenger cells are part of our immune system that will get rid of those cells and reuse its parts and so on. And so that's an actual picture of that happening. And so what's going on inside cellularly, I said that it can be triggered by a ligand. So we have a couple pictures here I wanted to show you. This picture here shows you a receptor and some inactive proteins inside the cell. What happens is, remember, it can be triggered by a death ligand. So here's your death signaling molecule. When it binds to this receptor protein, it activates molecules, so now these CED4 and CED3 are activated, so that's the cascade, and you get other enzymes that are activated, and these enzymes start to break up the cell parts and forming those blebs, and eventually the cell will go again from looking like this to looking like this as a result of those molecules being activated. So that is a cell signaling process. Like I said, that can be a good thing in development or a bad thing. All right, and that is the end of this chapter. Come to class with any questions that you may have or think of.